Welcome to our webinar entitled Targeting VPS4A and B Synthetic Lethality, a New Tool for ATPase Drug Discovery. I'm Matt Blackburn. I'm an application scientist at BellBrook Labs. I completed my PhD training at the UW-Madison in biochemistry with a focus on P-type ATPases. Today, I'm going to be going through the same webinar that we presented on August 12th. This is a recorded version, which will be accessible to everyone at your leisure. Okay, so let's get started. Very briefly, what we will talk about today is a brief background on the target enzymes here, so VPS4A and VPS4B. I will describe their biochemical activity in vivo and in vitro, and then describe how we have used Transcreener to build an assay around this. I will describe our assay schematic, and then how we have developed a specific ATPase assay using the ADP² fluorescence polarization assay. But I will also describe the different assay formats that we can use to do the same type of experiments. I will then talk about the screening data that we generated, doing a pilot screen for compounds that potently inhibit VPS4A, and then how we did a selectivity profile to detect off-target effects in VPS4B and other related AAA ATPases. Finally, I will summarize what we talked about, and then I will review the collected questions that we recorded during our Q&A. Okay, so let's get started. In brief, vacuolar protein sorting associated protein 4, VPS4A, or VPS4, has two isoforms, A and B, and they are members of the meiotic clade AAA ATPases, which is a large family of ATP hydrolyzing enzymes. The full name is ATPases associated with diverse cellular activities. VPS4 is crucially important for the escort system, specifically its role in living cells is to recycle by remodeling or disassembly components of the escort system. Specifically, it returns escort 3 polymers or individual components of escort 3 back into the cytosol. And this activity is associated with many membrane remodeling events within the cell. And this can be anything from vesicle formation, multivesicular bodies, cytokinesis. It's also implicated in viral budding and autophagy. Of course, when proteins are maturing in the ER and in the Golgi, escort is important for that process as well. Without this final stage of recycling, the entire system can break down, which is why it becomes important in disease with phenotypes that reflect a disordered escort system. Isoforms A and B, importantly, share 80% amino acid sequence identity and have high structural conservation. Now, what makes VPS4A and VPS4B of clinical importance is that frequently in cancers, VPS4B is deleted. And so typical of cancer, where you have these genomic instabilities that result in large-scale deletions or fusions, you end up with a pathway addiction. And this is sort of the basis of synthetic lethality. So in a case where you have cells or animal models where VPS4B is deleted or deficient, you start to get dependence on the other isoform. And when you end up abrogating or deleting VPS4A expression in a VPS4B negative background, you induce pro-apoptotic signaling. You induce inflammatory cytokines and anti-tumor responses in quite a significant way. But beyond cancer itself, beyond this pathway addiction aspect, this is also important for diseases that are related to membrane remodeling. Again, these are for this reason, this is a therapeutic target in, viral infections where the membrane forms the outer envelope of enveloped viruses. And for neurodegenerative disorders. So neural pruning, for example, in healthy cells in healthy animals, is also a process that relies upon a functioning escort system. So in vivo, the biochemical activity of VPS4 is actually characterized by oligomerization into higher order complexes. And typically, VPS4 adopts a hexameric configuration, sort of in the shape of a wheel with a central pore. And what this configuration relies upon initially for assembly and stabilization is recruitment of the protein cofactor VTA1, which then promotes binding of ATP. And together, these two binding events lead to stabilization of that higher order structure. And then you gain, and then the entire oligomeric complex becomes competent for ATPase activity. As we see in the reaction below, the reaction schematic below, of course, this higher order oligomerization is not strictly necessary for ATPase activity, but it does promote it. And with this hexamer, this ATPase activity is coupled to the recruitment of escort-3 components that are associated with the membrane around the neck of a budding vesicle, for example. And they are essentially peeled off the membrane and threaded or extruded through that central pore. And that is the mechanical basis for how escort-3 components are recycled back into the cytosol for rebinding as the neck of the vesicle continues to narrow. Now, in our in-house, when we've built this assay, our Transcreener ADP² assay around this activity, we target only the ADP generating capacity of this enzyme. And consistent with the goals of a lot of pharmacological interests, we are looking to characterize the ATPase activity so that we can then screen for ATP competitive inhibitors. And the way this ends up working with our assays is we will have recombinant purified VPS4. We then will build a reaction mix containing a concentration of ATP, which we have predetermined from KM analysis, which I will get into in a couple of slides. And then we will run the reaction, and then this is where Transcreener comes in. We build a detection mix containing an ADP antibody bound to an ADP molecule conjugated to a fluorescent tracer. And so what ends up being the readout is displacement of that ADP conjugated tracer by newly formed ADP produced by ATP hydrolysis activity of the enzyme in question, in this case, VPS4. And as ADP evolves into the enzyme reaction mix, it will displace that tracer in a concentration-dependent manner and result in a change in the fluorescence signal. And what this looks like at BellBrook through our assays, this can take several forms. This readout can take several forms. We offer essentially 3 formats for directly measuring the generation of ADP. And in the first case, we offer a fluorescence polarization assay. This is probably our conceptually simplest assay. Essentially, the small molecule of the tracer, that is the tracer, bounds to the much larger molecule that is the antibody, exhibits polarization. And when ADP, when it gets kicked off of that antibody, it is able to tumble more freely in solution, resulting in a loss of polarization. So displacement leads to a decrease in the total polarization of the signal. And that is what we measure through the FP assay. In the second instance, we have a TR-FRET-based assay where we have, again, an ADP antibody, but this is conjugated to a terbium donor. And when the ADP tracer is not yet displaced, it is close enough to this terbium donor that it is able to undergo Forster resonance energy transfer, and it will emit its own lower energy wavelength that it as a lower energy photon that has that is this excess energy that it has absorbed from the terbium donor. So a typical FRET. And so again, in this case, we were measuring a decrease in the signal. In this case, the total FRET signal, reflecting that proximity of the acceptor, the tracer, to the donor, the terbium bound to the antibody. And then finally, we have a fluorescence intensity assay. And this is similar to the FRET assay in that it is proximity based, reflecting a change in reflecting a change in the fluorescent signal. In this case, the antibody is bound to a quencher, where in a proximity dependent manner, it is absorbing the total fluorescence coming off of that tracer. That ADP tracer that is bound to the antibody. Displacement then leads to that tracer being freed from this quenching antibody, and it reflects a total evolution of fluorescence in the enzyme reaction corresponding to the formation of ADP. In each of these cases, this is direct detection of ADP as it becomes increasingly concentrated in the reaction mixture. The advantages of Transcreener are actually quite significant. These seem at face value like they could be complicated assays, but they're all very simple to set up, very simple to measure, and they're all very reproducible. Most importantly, as I've mentioned, this is a direct method for homogeneous detection of ADP. It is also, we like to advertise that this is a single edition mix and read format, and that is true. What you end up doing with our assay is you build your enzyme reaction first, and then our assay comes into play where you build a detect mix that only has 3 components in it. We have a buffer that we build the components into, and the other 2 components are the ADP antibody and then the ADP tracer. So these 3 components are combined. We describe how to do this in our technical manuals. And then at the end of your enzyme reaction or during the course of it, you can just adjust the buffer to exclude the quenching agent, which here is typically EDTA. And you just add this detect mix directly to your enzyme reaction, and then you watch the displacement take place, measuring it directly. Importantly, we build these assays to be amenable for any high-throughput screening application. And in-house, we have validated them to work with 96 well, 384 well, and 1536 well plate formats. Additionally, another crucial advantage of these assays is that all of these readouts, whether it's the fluorescence intensity, the fluorescence polarization, or the TR-FRET, they all use far red fluorescent tracers. And because far red wavelengths are really, really rare in nature, you're going to essentially just by default exclude a lot of noise associated with scattering or autofluorescence, which tends to be in the more green range. Finally, these assays are built to be highly sensitive. we design them to exceed a Z prime factor of 0.7 at 10% substrate conversion, which is that range where you're still in that initial velocity of the reaction. And it can also accommodate a wide dynamic range of ATP concentration, ranging from 0.1 to 1,000 micromolar. And this makes it up to 10 times more sensitive than competitor assays, which of course also tend to have many more steps involved. Because we have these fewer steps and reagents in the transcreener assays, this also minimizes the amount of handling by the user that has to take place, which inherently will make it more reproducible. And it will also minimize interference from assay components because there's so many fewer of them. You don't have to worry about doing multiple conversion steps to detect the product, for example. And those are all, each time you introduce a new component, you run the risk of introducing this kind of interference that will affect the fluorescence signal. We also have validated these detect mix components to have high DEC stability. And this is really important for customers who may be, and users who may be screening at really large scales, dozens to hundreds of plates in a 24-hour span. When you're doing that many plates at once, you're going to really want to make sure that your mode of detection is unaffected in a time-dependent manner. You want your results to only reflect the enzyme activity instead of something happening with the detect mix that is unknowable. So we have validated these to be stable in excess of 24 hours. And finally, because these assays are built to detect ADP itself as it forms in solution, it is essentially universal. It can be used to detect any ADP-producing enzyme, including the more than 500 known kinases in the human proteome and the hundreds of other ATPases as well. And here I show a selection of applications that we have developed in-house, where we have validated these assays on specific enzyme targets that are of great therapeutic and clinical interest, including P97, which is extremely important in cancer research and therapeutics, but also VPS4A and VPS4B, as well as several SMARCA isoforms. Shown kind of in the middle of that column. A little bit more on the advantages of a transcreener. I alluded to this on an earlier slide. You can run this assay in endpoint mode, where you quench the reaction after a fixed period of time and then measure the total production, or you can build a detect mix that excludes a quenching agent. And essentially measure the formation of ADP over time in a kinetic manner. Importantly, an application of this is that you can perform drug residence time determinations using the classical catalytic jump dilution. And here I show on the right an example of a residence time determination for inhibitors of EGFR. Using the product formed and the integrated rate equation. That is the basis of the drug residence time determination. Of course, this is a really important piece of information. It is becoming increasingly acknowledged that drug potencies are not sufficient for describing the total pharmacological activity of a drug. And that is because as these assays tend to be performed in systems that are much simpler than the ultimate target system, a human, for example, you will tend to have a lot of variables that are not accounted for. And what drug residence time specifically accounts for is not just the tendency of the compounds to bind the target, but also how long it will occupy the target enzyme. And this extra piece of information is really important for determining the lifetime of a drug, the pharmacological lifetime of a drug in the system that it will be used in. And it can also be used to determine the likelihood of off-target effects that potencies may not be as clear in identifying. Okay, so that comes to the second part of the talk. Here I'm showing an ATP-KM determination for VPS4A. This is an important early step in determining the enzyme parameters for VPS4A as we progress towards doing drug screening and follow-up dose response assays. So as I mentioned earlier, when you are screening, when your goal is to screen for ATP competitive drugs, it is really important to know the KM of ATP for your target enzyme. And the reason is when you are actually doing your screening, you're going to want to do this at ATP concentrations at or below the KM. And the reason for that, of course, is, as I'm sure many of you know, if you have ATP at a saturating concentration with respect to the enzyme activity, it's going to artificially introduce competition with the very compounds that you are trying to measure potencies for. And it will immediately interfere with your experiment. So you want to make sure that your enzyme your enzyme reaction is running at or below this Km for ATP. And so here, as I show on this slide, we determined the Km for VPS4A for ATP to be about 36 micromolar. And in successive experiments, we ended up using well below that, 2 micromolar, to perform follow-up enzyme titrations. And the enzyme titration, of course, is going to be important for determining the range of enzyme concentration where you're going to be in the zone of about 10% substrate conversion, so that you're reflecting that initial velocity-like state. And that's going to be in the zone of activity where you're going to want to do your drug screens. So here I show titrations for both VPS4A and VPS4B using the information gleaned from the VPS4KM determination. All of these assays that we offer are based on displacements. All of these assays, I should say, in the Transcreener platform are based on displacement, and because of that, you get this sigmoidal curve. So to determine ADP converted by the activity of the enzyme, we also will include a standard curve that is built using the working concentration of ATP in the actual enzyme reaction. And once you have this standard curve, you can then convert that raw fluorescence signal, that readout, into ADP converted. And you can see over on the right here that once you do this conversion, you find that you have a direct linear relationship between the ADP formed and the enzyme activity. Okay, so given those extra pieces of information, we found the Km for ATP, we found our working enzyme concentration that we want to use. We then proceeded to identify potential potential inhibitors for VPS4A. So we started with the TOCRIS 2.0 library, which is a screen of more than 1,000 compounds that are known biologicals. And we identified from within this set 15 likely hits. Over on the right of the screen, I also show a Z prime determination that was paired with this experiment. And we will do this with each of our in the later phases of our drug discovery for a given enzyme target, because we want to ensure that our assay is reproducible and reliable. And I mentioned the Z prime factor metric earlier. We prefer this for our own development, and we recommend our users do this as well. Z prime factor tends to be a much better report of the robustness and reliability of your assay. And that's because not only does it account for the difference between the signal and the mock reaction condition, it also accounts for that variability in the signal itself. And so this leads to it being a better metric for the reproducibility of the assay and how reliable it can be. So we always go for a minimum. We always strive for a minimum of Z prime of 0.7, and our assays are designed to exceed that where possible. But more often than not, we tend to get above 0.8. Okay, so having identified those initial 15 hits, we then screened these for interference with the assay. And I don't show this explicitly in these slides, but the way we screen for interference in-house is we will essentially set up dose response curves for each of the hits, but in a mock enzyme reaction that reflects 10% ATP conversion. So in the example of VPS4A and B, where we use 2 micromolar ATP, this would be a reaction mixture without enzyme, but including the buffer and ATP and ADP at concentrations of 1.8 micromolar and 0.2 micromolar, respectively. So that's 10% of ATP converted into ADP. And so the way our detect mixes work, of course, is directly detecting that displacement by ADP. So in principle, if there is no interference with the fluorescence signal of this detection reaction, we should see a flat line. The dose response curve of the inhibitor in question should essentially be flat. Where it is not flat, we tend, if it is clearly interfering with the total signal of the assay, we will exclude that. And using that information, we excluded 3 of these compounds. Moving forward, of the 12 that we had validated, we picked 3 based on how frequently they have appeared in the literature as potent inhibitors of other assays, or in some cases even activators. And these are DBEQ, BVT948, and polygodial. And so we did follow-up dose response assays with each of these. And then again, with a paired Z prime factor determination to make sure that the assay is functioning to the standards that we expect, which it is. We registered a Z prime score of above 0.9. Reflecting that this assay is highly reproducible and robust, and that these results are likely to be reproduced. And on the following slide, we actually do show that when we move into a selectivity profile, where we bring in additional AAA ATPases. So those were dose response assays originally for VPS4A. This profile brings in VPS4B, catenin, spastin, and phygitin. And so each of these has a dose response assay for the 3 inhibitors that we originally did follow-up screens on for VPS4A. And just at a glance, you can see right away that these curves collectively reproduce that initial observation that DBEQ, for example, tends to be a less potent inhibitor for VPS4A. But we also see it's less potent in a few other instances as well. And then we see some higher potency inhibitors. It's higher potency inhibition by BVT948 and polygodial. This becomes clearer when we bring all the values together into a table. And here you can see immediately the DBEQ result reproduced for VPS4A, but also for VPS4B. You would expect that, given that those 2 isoforms are essentially functionally redundant. But also for other related ATPases, you see even lower potency, for example, for catenin, but much higher potency by comparison for spastin. But then when you get into these inherently more potent compounds, BVT948 and polygodial, inherently more potent, that is, for this set of enzymes, you can see other differences emerging. Catenin, for example, is much less inhibited, comparatively speaking, by BVT948 than the other four enzymes are. But on the opposite end of inhibition, we have spastin being the most potently inhibited. So that would reflect an important off-target effect. If you were trying to inhibit only VPS4A or VPS4B, you would have a lot more inhibition also happening with spastin. And so that's an example of how these data can guide structure-activity relationships when you're trying to build a new compound with really targeted specificity and potency for a single enzyme or a really narrow group of a really narrow class of enzymes without having a lot of undesirable side effects, which are clinical manifestations essentially of these off-target behaviors. You would want to look at these data and then use that to make adjustments to the structure of your molecule to try to get rid of these off-target effects and maybe enhance potency on your desired target. And then you would run the screen again and see if you were actually hitting that metric. And based on what this iterative cycle of experiments teaches you, you can narrow closer, you can get closer to your goal of designing a specific compound that will be of great therapeutic value. Okay, so that brings us to the end of the talk. Today, we talked about how we used the Transcreener ADP assay to do direct homogeneous detection of the ATPase activity of the AAA ATPases VPS4A, VPS4B, catenin, spastin, and phygitin. Across the board, this assay provided excellent data quality exceeding the Z prime metric of 0.8. And we had a good signal window in each of these assays. And so what this means is that this presents a really robust system for scaling to high throughput screening levels for doing large scale screens for new compounds against VPS4A and related enzymes. Pilot screens that we did in that manner identified inhibitors that for VPS4A and VPS4B are new inhibitors, and we determined IC50 values of that. We profiled those hits against those other AAA ATPases that I mentioned at the top. And broadly speaking, what this shows is that our assay platform can facilitate the rapid discovery and validation of biologically relevant inhibitors for VPS4A and other clinically important targets, and aid in the identification of off-target effects, which is crucial for guiding structure-activity relationship and development. So finally, in our Q&A session yesterday, we recorded the questions that were fielded. I will go through each of these today and describe both the question and the answer. So for the first question that we got yesterday, our questioner asked, can the platform be used with cellular assays? In principle, our assays can be used with cellular assays and with lysates. However, we do not recommend this. Our assays are designed and rated for use with purified components only. And the reason for that is that in a chemically undefined milieu, such as a lysate or a living cell, you cannot guarantee interference from contaminating nucleases, which would degrade the very products that we're trying to detect, or even in the case of ATP itself, it might prematurely degrade the ATP, which both of which will affect the assay window and its reliability. Additionally, there's just like a lot of other components in lysates that could introduce spurious fluorescent signaling. Excuse me. For example, lipids from membrane components tend to cause a lot of scattering and autofluorescence. And even though our far red tracer design is optimized to minimize that, introduce enough noise and it's going to affect your signal, period. So we do not recommend this for use with lysates. Our second question was, can this be used with other plate formats such as 1536? I mentioned earlier in the talk that our assays are built for 3 different plate sizes, 96 well, 384 well, and 1536 well. Again, we build these assays with the intention of them being amenable for high throughput screening scalable experiments. The only additional piece of information that I would add to this answer is it's important to use the correct materials, or rather plates that are made from the correct materials. Our assays are all fluorescence based, so you're going to need to use opaque plates that are low binding, square weld, which aids in automated dispensing, round bottomed, and the plate will need to be black in the case of fluorescence intensity and fluorescence polarization, or white in the case of TR fret. If you have trouble finding these plates, we also offer these as part of our product listing through an agreement with Corning. If you want to pick up some plates through us, just contact us about it, and we can send you these plates if you need them. Okay, so our third question was, I showed 3 transcreener fluorescent readouts. How do you choose which one is best for you? The short answer is it's up to you. This depends on the user's own experience, their own instrumental needs, and their goals. So, for example, in our case, we have instruments that can read the fluorescence polarization assay. And so with that barrier, I shouldn't say barrier, but with that instrumental requirement met, we actually prefer using FP in-house because it's just the quickest one. It's the simplest to set up, and it's quite robust. That being said, we also use the other 2 formats as well. We routinely validate all of these platforms. So in-house, we have the instruments to do all of these formats. But if you, the user, for example, are seeking higher sensitivity, you may prefer the TR fret assay. That's going to require different components than the fluorescence polarization assay. What the TR fret offers in terms of additional sensitivity comes from the time resolution aspect of the TR fret assay. And that is that gap of time between when the incipient excitation wavelength is cut, you have continued fluorescence by the terbium donor after that initial wavelength is cut. And so that is a relatively long-lived donor signal in the absence of the excitation laser, or sorry, wavelength after the fact. And so what that does is it continues to fluoresce after any kind of light scattering effects or other autofluorescence introduced by the excitation laser, sorry, wavelength. After that has all dissipated, you are left only with contributing fluorescence from the donor, which means your TR fret signal is going to be much cleaner. So this increases its sensitivity. And in other cases for fluorescence-based experiments, users may have more experience with TR fret or may prefer it for other reasons. So this is the assay for them. For fluorescence intensity, this of the 3 formats is the most amenable to any plate reader. Most plate readers will be capable of measuring FI. Unlike the other 2 assays, it does not require specialized optics. With the FP assay, you may need a polarizer. With both FP and TR-FRET, you're going to need filters. But with the FI, you can use just a monochromator because you can measure that bulk fluorescence intensity as it increases in the enzyme reaction. So again, to sum up, it depends on your needs and in your experimental goals. Our fourth question, we have tested a number of AAA ATPases so far. Do we have plans on validating more enzymes as well from this family? More broadly speaking, this kind of speaks to a larger question of what if I have an enzyme that I'm interested in and I'm coming to BellBrook, but I don't see it as an application on the page. What then? What I would say is contact us. We have a really robust services department where we can build a custom assay around your target. We can do all of that initial work for you, where if you have an uncharacterized enzyme, or one that is not well characterized, but is an emerging target in the literature, we can take a lot of that initial legwork out of your hands and sort of develop and optimize that activity for you. We can identify the ECs for you. We can identify the KCAT. We can determine the KM for ATP if this is an ATPase. We can get all of that set up and get you right up to the point where you're ready to start doing drug screens. We can even do screens for you. Basically, a lot of the work that you could do yourself, but perhaps you're eager to get further down the road faster, we can do a lot of that upfront work for you. So if you have an enzyme that you need to have validated or want to have validated and you don't see it on our page or on any other company's pages, contact us and we can build something for you out of that. Okay. Another question that we got is if we were to build a VPS4 assay like this in our lab, what steps should we take? The first thing you should do is make sure you have a plate reader that is equipped to read one of our assay formats, and that you are picking the format that is right for what you have and what you have experience with. And we can talk about that. You can contact us and we can guide you through that process too. You're also going to make sure you have the correct consumables, the plates themselves, for the assay format that you have chosen. You're then going to want to make sure within your given assay choice that your instrument's maximum signal window is determined before proceeding. And we have instructions for this in each of our tech manuals for how to do this. Essentially what this boils down to is you will take our detect mix and build mock reactions out of it. And in one case, you'll have a mix containing only tracer or rather tracer in the absence of antibody. And what this will give you is essentially one extreme end. If we're talking about fluorescence polarization, for example, that free tracer mix will reflect the minimum polarization signal. And so in the case of measuring an ADP assay, for example, that would reflect the maximum detection limit because it reflects maximum displacement. On the other mix, you will have antibody included, so that all of that tracer would be bound, and so that would be the opposite end of the assay window. And you want to determine that signal range so that you actually know that you have confidence that successive measurements are giving you that are not being limited by an incorrect instrumental parameter, for example. Finally, when you're actually getting into your enzyme experiments, you're going to need to start with enzyme titrations. You need to find out, does my enzyme have activity? And then if it does, what is its EC50 or its EC80? Do I need to modify the buffer, for example? to enhance the activity? Is it a very slow enzyme? Would it benefit from a 37-degree incubation versus a 30-degree incubation? Does it need metals like magnesium chloride, or I should say divalent magnesium? Might it benefit also from including manganese? So in the case where you're optimizing the buffer, you may want to do a buffer matrix where you have multi-parameter or multi-component titration going on in the same experiment, where you're varying all of these different components, introducing them, but varying their concentrations all at the same time. And you can get a really broad picture of what likely combination of components is the most beneficial for your enzyme. And then follow-up titrations based on that selection will help you narrow down your optimal concentrations. If your enzyme's activity is satisfactory already, you may not be interested in further buffer optimization, and you may be ready to move forward with determining your enzyme's kinetic parameters, your Km, for example, for ATP, if you're going to be screening for ATP competitive inhibitors. Your enzymes EC50, EC80 at your currently optimized concentrate or optimized conditions. And those pieces of information are going to guide how you build your pilot screens. And so from there, then you go on with your dose response assays and so on, as I showed in the talk. Okay, we're getting to our final two questions. The next question was, what is the maximum ATP concentration that may be used with these assays? The maximum concentration is 1,000 micromolar or 1 millimolar ATP. Our final question was, are there assays available for other nucleotides such as GDP and UDP? Yeah, we offer many variations of the Transcreener assay. Today we talked a lot about ADP. It was one of our earliest assay formats developed, but we have a lot of other platforms for other types of products. So other types of enzyme products that are generated and can be directly detected. So we have assays for GDP detection. In the case of GPCRs, for example, for glycosyl transferases, of course, yes, we have kits, we have assays for that to detect UDP generation. We even have exonuclease assays, which currently detects DAMP. But we also have phosphodiesterase activities, which we can detect, which focus on the generation of AMP and GMP. I would say for more information, it would take a long time to go through all of them. But for more information, if you don't know where to begin, you can go to our homepage, bellbrooklabs.com, and at the banner at the top, there is a tab that says Assays by Target. If you click on that, it'll bring you to our applications summary page, and this shows you all of the types of enzymes that we currently have developed assays for. And this will include everything from your GDP generating enzyme, your UDP generating enzyme, your DAMP, and so on. We also have a lot of other assays that target other products besides nucleotides. So I would recommend go to that page if you don't know where to begin, or if you're curious about what we offer. That's a good place to start. Okay, that is all the time that we have for today. Thank you for attending our webinar on August 12th. Thank you for your questions. If you have additional questions, please feel free to contact us via email at info@bellbrooklabs.com. Again, that is info@bellbrooklabs.com. This webinar will be uploaded imminently, which you can review at any time is convenient for you. Visit us at any time for our latest developments. We update the site frequently with new posters, with new application notes. and occasionally new publications. So check back on us from time to time. We're always generating something new. And that's it. Thank you again. My name is Matt Blackburn. I'm an application scientist at BellBrook Labs. Feel free to reach out with any questions or concerns. We're happy to talk.